Sir, let us welcome the guest by welcome note by our principal, sir. Sir, over to you, sir, Natas.
Thank you, Mr. Kranti. Respected today's speakers, Professor Suresh Babu and my fellow teachers and my dear students, my warm welcome to one-week online FDP SDP program entitled Recent Trends in Pharmaceutical Innovation and Research from 22-24 November 2023 on behalf of students, staff and management of Vishnu Karga Pharmacy. The main objective of FTP is to update you the information about the technology-driven pharmaceutical innovation and how the emerging technology helping in pharma innovation. My sincere thanks to all speakers for acceptance of our invitation in spite of their busy schedule. I wish you all the best to the faculty and students. Make use of all the sessions. Today, one of the eminent Professor K. Suresh Babu will enlighten all of us about how the natural products discovery. Okay, so today you will be many takeaway points you can find today's talk. Thank you all and over to Kranti.
Thank you, sir. Now I request Dr. Kiran Kumar to introduce Suresh Babu, sir.
Thank you, Kranti sir. It's my great privilege and honor to give the introductory note about today's speaker and eminent personality in the research area of natural product, Dr. K. Suresh Babu sir. Dr. K. Suresh Babu sir obtained his master's in 1999 and doctorate in natural product chemistry in 2005 from Indian Institute of Chemical Technology, Hyderabad. Later on, he moved to pursue his postdoctoral research at National Center for Natural Product Research, University of Mississippi, and he returned later. After that, he returned to India and joined in CSIR service in 2007 at Indian Institute of Chemical Technology, Hyderabad. His research group has successfully isolated several new bioactive molecules from natural sources, and he published 200 papers in essay journals, and he filed 12 patents of India, PCQ, and US. He is leading many projects of national importance in new drug discovery, standardization of herbal drugs of commercial importance. His research interests are new drug discovery from natural products, herbal drugs, and their standardization, botanical pesticides, chemical modification of lead compounds, and their SAR studies. And he is a recognized research guide for doctoral and postdoctoral programs, and 5 scholars are pursuing under his able guidance, and 25 scholars got the PhD degree. For his great research contribution, he received several awards and honors. He received Young Scientist Award from Organization of Pharmaceutical Producers of India in the year of 2010. And he received Young Scientist Award in the year of 2010 from Andhra Pradesh Academy of Sciences. He received Best Performance Award for the year of 2011, conferred by Director ISCT Hyderabad. Elected as an Associate Fellow of the AP Academy Sciences in the year of 2012. He is a scientific consultant to MS Himalaya Healthcare R&D Bangalore in the year of 2015 and 16. Elected as a Fellow of Association Biotechnology and Pharmacy in the year of 2016. He is a editor board member of several journals like Pharmacognosy Magazine and Asian Journal of Chemistry. He elected as a Fellow of AP Academy Sciences in the year of 2018. And he received Dr. A.K. Singh Memorial Young Scientist Award for the year of 2019. Elected as a Fellow of Telangana State Biodiversity Board, Hyderabad, Telangana. And his 2 patents were commercialized to Swami Labs PVT Limited, Bangalore, and those products are now available in international market. And he transferred 2 process technologies to the several companies like Amaya Life and Wellcare Life and SDG Healthcare. These products are released in March 2023 and available in the market. Thank you. Thank you, Kranti sir, for giving this opportunity.
Thank you very much, Kiran sir. Now I request today's guest to deliver his valuable research. Sir, over to you, sir. Suresh Babu, sir.
Yeah, thank you, Principal Garu. And thank you, Kiran, for your good words about my introduction. And once again, I would like to thank the organizing members for giving the opportunity to share some of our views regarding the natural product research, where we are working in this area from the last 2 decades. Am I audible, sir? Sorry. Hello?
Excellent, sir. Excellent.
Am I audible? Is it okay?
Yes, sir. Excellent. Excellent.
Right. Thank you. Today, just I'm going to discuss about the recent approaches in natural product research for drug discovery. My slides are visible?
Yes, sir. Excellent, sir. Excellent.
Okay. So, since we are working in this area from the last 2 decades, we are adopting several approaches in the natural products, including their standardization and then making the natural product lead as a drug candidates. So today I will share some of our recent work also in this platform. So slides are not moving. Okay. So before going to my presentation, just it's a customary to tell about our institute. It's Indian Institute of Chemical Technology, which comes under CSIR network, which is a Council of Scientific and Industrial Research under the CSIR government. CSIR, it is under the Ministry of Science and Technology, Government of India, established in 1942. It's an autonomous body and has the largest scientific body. And in the entire India, we have almost 37 laboratories under the 5 disciplines. Color indicates the discipline: biological sciences, chemical sciences, and engineering sciences, information sciences, and physical sciences. Coming to Hyderabad, we have 3 laboratories in Hyderabad, one NGRI, CCMB, and ISCT. NGRI belongs to physical sciences, CCMB belongs to the biological sciences cluster, and ISCT belongs to chemical sciences cluster. When coming to ISCT, ISCT is established before India's independence, erstwhile Nizams, in 1944. After the independence, it has been taken over by the Government of India and then named as a Regional Research Lab, Hyderabad, RRLH. So in 1989, the name has been changed to Indian Institute of Chemical Technology based on the development of the several technologies and transfer to the pharma, chemical, biotech industries under the able leadership of Dr. A.V. Ramarao. At that time, in 1989, several technologies, particularly monochromatophos and chlorifier phos technology has been developed, and then the cost is almost reduced to half of the price. So based on that great achievement, the name has been changed to Indian Institute of Chemical Technology from Regional Research Lab, Hyderabad. Since it's a CSIR network, so in 2012 CSIR has been added before the name of IICT. So since then IICT is serving the chemical industry and the society from the last 80 years. So these are major expertise and divisions in IICT. Starting from major strength of IICT is a chemical sciences or chemical technology, chemical synthesis, and of course we also have the supporting of state-of-art facility of analytical equipment like NMR, MAS, XRD, and other hyphenated techniques, HPLC. Of course, we also have Center of Excellence of Polymers and Materials, Molecular Modeling, Pharmacology, Toxicology, Chemical and Design Engineering. So these are Catalysis Division, Genetic Engineering Laboratory, and Diversity-Oriented Synthesis Lab, Fine Chemicals Lab, Agrochemicals Lab, Pheromone Lab, Natural Products Laboratory. So major strength is the development of the drug process for the drug intermediates and development process for the drugs, and also commercialization of those processes to the industries. And recent achievement that we have developed a technology for process technology for the adjuvant, where it is used in the Covishield vaccine. So every Covishield vaccine contains a chemical which is called adjuvant that has been developed by the CSIR IICT. Similarly, in the lipid science division, we have developed, IICT has developed almost majority of the technologies in India now. Presently, almost 70 to 80% industries are utilizing the IICT process technology for the production of the rice bran oil. So these are the few achievements of IICT. And coming to the drug discovery, if you take any drug, definitely there will be origin of traditional medicine or natural products or folklore medicine. The traditional medicine basically it refers to a medicinal knowledge and innovations and practices of indigenous and local communities around the world. So in the ancient times, our ancestors used it to identify the plants, surrounded plants as a medicinal plants and toxic plants. These knowledge has been rooted to their generations together and then formed in the part of their culture, and then as it is established as a traditional medicine. We can call it as an Indian traditional system of medicine and Egyptian system of medicine, China, all these systems of medicine has been formed like this. So this knowledge has been developed, experience gained over the centuries, and of course transmitted orally from one generation to other generation. The traditional medicine in India can be codified into classified into two categories. One is codified, which comes under Ayush Ministry, Ayurveda, Unani, Siddha, and Homeopathy, and non-codified folklore medical system, where there is no scientific sanctity, but still the tribal people are utilizing the system for their health care. And of course, these both systems are contributing equally to the primary health care in India. So this traditional medicine contributing to the humans in the from the last more than 4,000 years ago. But only thing is the major hurdle is quality control aspect. But in this present scenario where we have the huge technology and of course analytical techniques, hyphenated techniques, so we can do much better in this present scenario. As I told you, this traditional medicine is also utilizing in the developed countries under the different name, complementary and alternative medicine, where this is available to the public, common public as a standardized medicine and over to the counters, OTC counters. And here I gave some pictures in different countries. And this type of people, in particularly in India, if you visit the tribal people or rural areas, or majority of the tribal people or rural people are still depending upon this folklore medicine, and even though they do not have any medical degree, RMP, PMP or MBBS, but they are surviving based on their traditional knowledge which has been gained from their forefathers and generations together. So these people are named as a traditional medicinal practitioners. So recently, Government of India also conducted a survey regarding the existence of these people for identification, and almost 4 to 5 lakh people are existing in the entire India, distributed north to south and east to west. Particularly, majority of the tribal people will utilize this traditional medicine, and of course, in India, we have the heterogeneous group of tribal people when compared to other countries, and particularly government, our constitution recognizes 212 tribes around the different parts of the country, Himalayan region, Northeast region, and South region, Western India, and Andaman and Nicobar Islands. So based on this peripheral data, World Health Organization conducted a survey to know about the facts of the utilization of the plant-based medicine for their health care around the globe. So it has revealed that more than 80% of the world population relied based on the plant-based traditional medicine for their healthcare needs, and further it has also listed another 21,000 medicinal plants around the world. And when coming to India, where India is known for botanical garden of the world, and we have very distinguished 16 agroclimatic zones, and majority of the plant species, like 45,000 different species are existing, and majority of those species are utilized in our system, traditional system of medicine like Ayurveda and other systems of medicine, and almost another some 5,000 products were included in the pharmacy of Ayurvedic treatments. These are the major natural sources: plant kingdom, microbial world, animal sources, marine world, and venoms and toxins. So among these 5 sources, plant kingdom has served a lot in the form of the Ayurveda and other kind of medicine. And microbial world, best example is penicillin, which has been derived from microbial world. Likewise, if you see the animal sources, some of the drugs which are in the present market also derived from animal sources. Best example is epibadrins, which are derived from the frogs. Similarly, marine world, antibiotics and anti-cancer drugs which are in the present market are derived from marine world, and some drugs also derived from venoms and toxins. And the lichens, this is also one of the important natural sources, but not explored chemically for their therapeutic usages. And lichens also known from ancient times for their therapeutic properties, and which has been utilized in the ancient times. Basically, lichens are a symbiotic organism of fungi and cyanobacteria. They are known for their antioxidant, cytotoxic, and other activities. So these are different applications of natural products, starting from therapeutic application, artemisinin, where it is a great drug which has been discovered by Professor Yo-Yo Tutu, Chinese professor, and further she has also been rewarded the Nobel Prize. And coming to ivermectin B, which is very well known as a crop protectant, rosmarinic acid, food preservative, and epigenin, which is very well known in beverage industry as a colorant, linalool, perfume industry, stevia artificial sweetener, and linoleic acid, very widely used as in the perfume industry, as in the lotions particularly. So these are the major applications. All these molecules are very well utilized in our daily life, and very demand in the industries also of different, starting from herbal medicines to cosmetic, pesticides, and other pharmaceutical applications. Recently, of course, this is one of the example, QS21, which is a part of which is very well known as the adjuvant and part of the many vaccines, including Covishield and other vaccines also. This is the only source from the natural product. There is no synthetic source of this compound. And why natural products means natural products are traditionally established from generations together, and there is a long history of their utilization from starting from almost 2600 BC, where there is evidence of used by the Mesopotamian civilization. And when coming to Egyptian civilization, it has also there is evidence 1500 BC. In that era, almost 700 drugs were documented. And when coming to famous Chinese medicinal system, and almost 1100 BC, dated back 1100 BC, and majority of the drugs has been documented in those days. And these are traditionally established as in the form of traditional system of the medicine of each country. When coming to our country, Ayurveda which is a very long established system of medicine practiced in India from thousands of years, and now it is getting worldwide attention as an alternative system of medicine, particularly in the post-COVID period. People are more interested to take plant-based medicine instead of the allopathic medicines. Of course, the Ayurveda and other traditional system of medicine do not concentrate on the single compound based because these are all holistic medicine, but these system of medicine like Ayurveda, other system of medicine made a base for the discovery of the several new drugs in the modern medicine. Here I just quoted some of the drug leads were extensively used in the formulations and other drug properties, like Rawalpia alkaloids, which are derived from Rawalpia serpentina to reduce hypertension. Soralin is very well known and derived from Soralia corifolia, very well known for in vitiligo, and olarihan alkaloids and piperidines, picrosides, phyllanthines, and vithanilides, steroidal lactones, and glycosides. And these are all economically affordable, and of course, procurement of raw material also easy because widely available, and some of the plants also, cultivatables plants also, and these are very effective in chronic diseases. Coming to the toxicity, these drugs should be taken in the optimum quantity, then only these are effective. But otherwise, if you take more quantity, then toxicity may adverse effect will happen. So, in the recent survey, particularly in the COVID 1 and 2 period, there are two universities, one from Pune and one from Mumbai. They also conducted a survey in the entire India, starting from Kashmir to Kanyakumari, regarding the consumption of spices versus the viral load. So generally, our consumption of spices is a part of our tradition. So we generally consume a lot of spices in our daily life by knowingly, unknowingly, starting from pepper and ilachi, mirchi, ginger, garlic, and the star fruit, lavanga, and all these general spices. Every kitchen hold is having these type of spices. So their survey revealed that if you consume more spices, then you will have the immunity. And of course, in the Lakshadweep people, Lakshadweep Island people, they have consumed a lot of spices. That's what the survival rate of COVID-19 and is very high, and even death rate is also very low compared to the main land people. And I also strongly believe that when compared to the Western countries, our country is the largest country, having almost 140 crores people population, and we are more than here. We have survived because of our tradition, like consumption of spices. That is only survived the India to not to big loss by the COVID-19 when compared to the Western countries, where in Western countries consumption of spices is very low. So this also revealed a eye-opening theme, because spices contains the polyphenolic compounds and anthocyanins, which are having the adjuvant properties. Adjuvants are nothing but immunity responding agents. So this is also one of the good research area to work on the adjuvant properties of the spices. So similarly, when coming to our biodiversity, almost among the 34 megadiversity hotspot, our country hosts three hotspots: Indo-Tibetan region, Himalayan region, and the Western region. Almost in these three regions, there is a lot of medicinal plants which are well utilized by the tribal people for their primary health care, and some regions that is also became a livelihood for the tribal people. So these three areas, there is a lot of medicinal plants which are not even identified taxonomically, but there is a huge opportunity to work on these medicinal plants, particularly in the Northeast region and the Western Ghats, and of course in some of the areas of Eastern Ghats also. And this is the drug discovery process from the natural products, starting from bio raw material to drug candidate. Starting from bio raw material may be plant, fungi, insect, and marine and microbes, whatever it may be, different natural source. Once bio raw material identified, it will be processed to prepare the extract. And there are a lot of extraction techniques are available, but based on the compound nature, the extraction techniques will be adapted based on oxalate, countercurrent extraction, and ionic liquid extraction, pressurized liquid extraction, solid phase extraction, supercritical carbon dioxide extraction, So based on the compound nature, these techniques will be adopted. Once extraction is prepared, it will go to the bioassay fractionation, that is using the chromatography techniques. Again, lot of chromatography techniques are available, starting from gradient chromatography, preparative chromatography, column HPLC, UHPLC, counter current column chromatography, supercritical carbon dioxide chromatography, membrane filtration, reverse phase chromatography, Cepheid X. There are a lot of techniques are available, but based on the compound nature and their polarity and their binding, the techniques will be adopted for the purification of those compounds. And once compound is purified, again it has to be characterized by the application of different spectral techniques. So here also there are a lot of spectroscopy techniques available from UV to X-ray. So every technique has its own significance for the characterization of the structure characterization of the compound. Once it is characterized, then that has to be a lead compound has to be prepared in the large quantities, where medicinal chemistry approach will come here for doing the depth biological activity studies. So once it is prepared, scalable and diversity synthesis, after this it will go to the different phase studies, phase 1, phase 2, phase 3. Once it is passes through all the, it will become a drug candidate. So almost starting to last, starting bio material to drug candidate will take 10 to 15 years if everything goes on well. And of course, it's a starting, it is a costly affair. That's what now majority of the companies are not coming forward to take the drug discovery from the natural sources. These are different 3 key steps involved in drug discovery of natural products: identifying the hits to lead and lead optimization. So if you see the contribution of natural products from the last 4 decades, and you can see almost 2,000 drug candidates were marketed, introduced in the market as a drugs. It may be either pure natural products or its derivatives. Majority contribution goes to anti-cancer drugs and anti-hypertensive drugs and antibacterial drugs. And when coming to the natural product activity, because it's a completely multidisciplinary activity, starting from the folklore healers, knowledge, folklore healers, Ayurveda, and taxonomy, and of course sociology, cancer biology. So it's a completely interdisciplinary activity, and of course it's a multidisciplinary activity, which involves the basis of the folklore medicine and further chemistry and then further biology and then further in neurochemistry, it is almost a validation of the folklore medicine. That's what multidisciplinary activity. When coming to our own contribution in the field of natural products, as I mentioned in my in starting, that we have been working in this area from the last 20 years. As part of our studies, we regularly visit the visit the rural areas and interact with the folklore people or tribal people where they are practicing the folklore medicine, to understand their knowledge and identify the potential medicinal plants for where they are using for different targets. So we are really very surprised while interacting with the tribal people based on their knowledge. And once the plant is identified, we will come to the lab and then go to the literature database, and once whether it is that plant has been scientifically validated or not. If it is not validated, we will take up the challenge and to work on that and identify the active compound or active fraction which is responsible for that activity. And once it is compound is identified, we will also involve in the chemical modification or chemical simplification of that molecule for enhancing the activity or reducing the toxicity, because majority of the times pure natural products may not having that much potency, and that's what in the present market majority of the natural products derivatives are became as a drugs. And once it is also studied the lead optimization and the SCR studies, and these are some of the targets, anti-cancer, anti-diabetic, and other targets we are working. When coming to applied research, we are actively working in the standardization of the multi-herbal formulations based on the marker compounds and process development of the active phytochemical ingredients, which are industrially viable process, and different natural products related projects based on the government funding. There are different approaches are available in the natural products, basically bioassay approach, random approach, and hyphenation technique. and phytopharma approach. And of course, among this bioassayer approach is a very famous one, as majority of the drug candidates has been discovered based on the bioassayer approach, which involved in extraction, screening of various targets, fractionation, and screening of fractions for biological activities, isolation and active compounds from the active fraction, and of course, characterization of the isolate by 1D and 2D NMR techniques. The random approach, which is quite opposite to the bioassayer approach, where because random approach is also equally important, because in the bioassayer approach, if bioactive compound is minor quantity and the inactive compound is major quantity, sometimes there is a chance of masking those active compound by inactive compound. In that case, that compound may be missed. So that's what we are also working on random approach, where you prepare the different extracts based on the polarity, and you isolate as many number of as compounds, and finally you screen for all the pure compounds for different targets. And hyphenation techniques, nowadays a lot of hyphenation techniques are available, starting from LCMS and UPLCMS, QTOF MS, triple guard, everything. And of course, molecular networking based approach also comes here, where our system is connected to the global natural product server. When you develop a total ion chromatogram, and it will be giving the known compounds, and it will be also giving the unknown information about the unknown compounds. And phytopharma approach, this is a basically a phytopharma approach is nothing but it's like an active fraction, which should contain at least 4 markers, and that 4 markers should be quantitatively and qualitatively assessed. And for every batch, the 4 markers should be the quantified. Sustainable quantity of these 4 markers should be essential in phytopharma approach. So I will explain each and every approach by giving some of the examples from our work. And here, this is the bioassayer approach. We have developed our own bioassayer approach, where it is involved in the active crude extract passing through the reverse space 18 column eluted with aqueous methanol and acetone to generate the 6 fractions starting from water to acetone. And all these 6 fractions are screened for biological activity. Once active fraction is identified, that active fraction is subject to column chromatography to identify the lead compound, and further the lead compound will be synthesized and for the larger quantities, and for in-depth biological studies and chemical modification will be done, mechanism of action studies and in vitro, in vivo studies. So this is one of the example of bioassayer approach, where it is involved in the isolation of the compounds from Piper species. As part of our program, we were involved in the screening of different Piper species, because as Piper species are more abundant of different species like Piper chaba, Piper nigrum, Piper longum, Piper beetula, Piper attenuata. A lot of species are available. As part of our program, we have screened for Piper chaba and Piper nigrum of cytotoxic activity as well as aldose reductase inhibition activity. Here, methanolic extract of Piper chaba showed potent cytotoxic activity, and the chloroform extract showed moderate activity with 22.5 aldose reductase inhibition activity. So with this active extract, we conducted the bioassayer approach to isolate the compounds. Here, from the methanolic extract, all the 7 molecules were isolated. Among this, the 5 molecules are new molecules, and the 2 molecules are known molecules. And here, after isolating the pure compound, these compounds were screened for anti-cancer activity against the different cell lines, Colo 205 cell line and other cell line. The compound 2 is showing almost most potent compound against the Colo 205 cell line, on par with the standard doxorubicin, and compound 3 showing moderate activity. And the structural characterization comes, we have characterized the structures of these compounds based on the 1D and 2D NMR techniques, and also finally confirmed through the mass fragmentation pathway, where we came to know that this molecule dimeric piperamide is forming based on the Diels-Alder reaction of the monomeric piperamides. So similarly, the active chloroform extract, we also conducted the chromatographic studies to identify the many number of compounds. Among this, the pipelartin IC50 showing moderate activity when compared to other molecules, and also the pipelat is isolated as a major phytochemical yield from this extract. So then we thought of making the derivatives around the scaffold as the majority of the natural product derivatives are became a drugs and also in the present market. So before going to the experimental work, initially we did the literature search to know whether what are the existing drugs in the market and what is the comparison of our molecule, and we came to know this diamide functionality seems to be the common in our compound and market drugs. So we thought of keeping intact of this diamide functionality and to work on this aromatic ring and double bond to form the derivatives. Initially we thought of doing the Michael addition reaction with different indoles, because indoles, we know very well, indole is a very good pharmacophore having a lot of biological activity. So for this Michael addition reaction, initially we did the optimization of the reagent and the solvent. After screening the several reagents, Lewis acid catalyst and iodine 10 mole % is optimized based on its yield, 80% yield, and solvent dichloroethane is optimized to selected based on its yield. Based on this optimization condition, we conducted the Michael addition reaction of pipelat with various indoles to form monomichael adduct and dimichael adduct. And here, these are the different Michael adducts we have prepared and screened for cytotoxic aldose reductase inhibition activity, and where we came to know dimichael adducts are showing very good activity when compared to the monomichael adduct. Here you can see derivative 3C and 3E showing activity almost 4 micromoles, which is equal to the standard drugs. The initial parent molecule activity is 160 micromoles, derivative activity is 4 micromoles. That is, the activity was enhanced 40 folds. Similarly, the modification of the aromatic ring, we have initially synthesized the pipelat, and further also we did the derivatives with various substituents in the aromatic ring, and these are the other derivatives. And unfortunately, these compounds are not showing potent activity, only moderate activity. So still we are working on the Michael adducts of pipelat to simplify the structure and also to improve the other further activity. These are another plant which is called Primula latifolia, which is also part of the Dashamola arista, that is a formulation of 10 plant roots. And this plant is not validated scientifically for their use. And we have taken up the challenge and then isolated 4 isotexane diterpenoids. First 3 compounds are isotexane diterpenoids and 4th compound is a dimeric compound. So all these 4 molecules were screened for anti-cancer activity, and almost all compounds are showing potent activity almost compared to the almost comparable to the standard doxorubicin. Since these compounds are scarcely available, very low yield in the plant, so for doing the biological activity studies, we need a more compound. That's what we, based on the reported literature and based on the known transformations, we have also synthesized the compound in multi-gram scale for the depth biological activities, and also to prepare the several of the derivatives based on the functional groups available. So these are the other kinds of molecules isolated similar way using the Bioseged approach, Primula tomentosa and Derris indica and Hedychium spicatum, Derris candens and Flammago capensis. So when coming to other approach, reverse Bioseged approach or random approach, here this approach is completely opposite to the Bioseged approach, where, as I mentioned, you just prepare the different extractions based on the polarity, starting from non-polar to polar, and you do the chromatographic studies for the isolation of the pure compounds as many as in various extracts. So we have taken Cypadasa basifera, where we collected this plant in the Rampachodavaram and Araku forest region, where we came to know this plant is utilized by the tribal people for their normal health care, and also in some of the places they are also using this plant for the agriculture purposes, insecticides. So we have taken this plant and prepared the various extracts and almost isolated 26 compounds, 26 compound limonoids, and almost 10 limonoids are new compounds and others are known compounds. All the structures of the limonoids were elucidated based on the 1D and 2D NMR techniques and also mass spectrometry studies. And these limonoids were screened for against different cancer cell lines. All these compounds were showing moderate activity. These are the structures of the other limonoids. And in some limonoids, basically Cipaferin, we have isolated in 500 mg and 1 gram level, where we have used that compound for the chemical modification, where we have done, we esterified the ester linkage and included some other functional groups to increase the activity, and it also showed good activity against the HEPG2 cell line, and further it is also showed some insecticidal activity against the Spodoptera litera. And this is also another plant where Saichandra chinensis, where it is available in the Himalayan region, and this plant is fruits are very famous in the Chinese system of medicine, and we have studied for their non-polar constituents and also the polar constituents, particularly lignans, and lignans are very high yield in nature. So we have also studied some the medicinal chemistry approach to add some of the pharmacophores to the lignans, and we succeeded. One compound showed very good activity, which is acting as a tubulin polymerization inhibition, gomycin B, and other derivatives also prepared. And these are the other drug leads where we have isolated and identified by using the random approach Burgenin derivatives, Burgenin and by its triazole derivatives and other derivatives, and the Caragana succensis in Nepal medicinal plant. Similarly, aloyamodin and isolated from the RMMOD, rhubarb and hedgerol, isolated from Hedicium spicatum and natural Aphonomyx polystachya, and some other lignans and Burgenin and epoxy azadirachtin isolated from neem oil, and Cipaferin from Cipadaceae basifera, and other limonoids from Trichilia conoroides. And the other compounds from Saichandra grandiflora and Stechospermum marginatum from the seaweed marine algae. And as I told you, lichens also important source for the therapeutic molecules, and where it was not explored much as part of their therapeutic usages. One of the important lichen is Usnea longissima, which is very well known from the ancient times and traditionally used for their centuries as a cardiotonic, stomachache, wound healing properties. We have studied this lichen, preparing the extraction and then fractionated into the 4 fractions using from hexane and ethyl acetate. And again, these 4 fractions were purified to identify and isolate the 4 compounds. And these are the structures of these 4 compounds characterized by the NMR and mass and IR studies. And this barbatic acid was first time we have developed a crystal structure for this barbatic acid, and all these compounds were screened for anti-cancer activity, and barbatic acid is showing very good very good activity against the 549 cell line. Further, we also studied flow cytometry analysis by using the cell cycle studies, and it is also indicating the G0, it is arrested the cell cycle in G0 by G1 phase, leading to the apoptic cell death, and further staining assay also suggested that induced cell death by apoptis, and immunoblot assay, capture 3 assay. Finally, it is also identified, it is also confirmed that it is showing the activity by cell death through the nuclear damage induced apoptis by the robust cleavage of PRP protein. Further, we also utilized the hyphenate techniques to identify the compounds based on the mass fragmentation. For initially, and we have developed the total ion chromatogram of the crude extracts, the Usnea longissima, and then identified those fragments. Finally, based on the MSMS fragmentation pathway, we have characterized the structures, and these are all the structures. Almost 40 compounds were characterized based on the MSMS principles, mass fragmentation pathways. Yes, and all the fragmentation rules are very much defined in the mass spectrometry, and we also proposed how these compounds are forming based on the observation of the key fragments in the MSMS spectra. So when coming to the other approach like a metabolomics approach or using the hyphenated techniques, LCMS based approach, generally in general approach, non-LCMS approach, generally we prepare the extract and do the fractionation, all the fractions, identify the active fraction, purified active compounds, and structuralization, activity confirmation, maybe new, non-new molecules. So this is a tedious approach, and of course and now a lot of hyphenate techniques are available. So by using hyphenate techniques, we can save the time, and also the accuracy is very high. And dereplication techniques, immediately you can also know what is the new compound, where is the unknown compound, by using the QT of MSMS and GCMSMS. So we have adapted this technique for the analysis of plant Trichelea corneroides, where it is also belongs to Malaceae family, neem family, where initially we have prepared the extract and injected the extract into the LCMS and to develop a total ion chromatogram of mass. And then by working in the different literature database, DNP, Reaxys, and we are able to pinpoint the new compounds and new molecular mass. Based on the new mass, we have done the preparatory HPLC to identify the target compound, and these are the compounds identified and structurally characterized by proton and C13 NMR. So similarly, we also applied the HRMS approach for the isolation of these 6 compounds from this lichen Ophiopharma ventosa of this color pigment. So we identified, and then further these are also tested against the B16 and DPPH and NBT SS. So all these compounds are showing moderate activity. So here further we also developed the comparative metabolite profiling of the constituents from different lichens by using UPLC MRM-MS approach, MRM multi-reaction monitoring method. At the same time, at a time, you can also quantify the fragment as well as molecular ion. So basically, this MRM approach is very well known in pharmacokinetic studies and metabolomic studies. And the first of its kind, we have adapted this technique in the analysis of the natural products, particularly in the lichens. What we have done is, lichens are very precious in the nature to balance the chemical ecology, and we have, and also these are very where these are grown in non-polluted area and moisture area. And once if you collect the lichen, and it will take another 3 to 4 years to grow itself. So that's what, not to disturb the chemical ecology, we have studied by using the LCMS approach, where we have collected the abundant lichen of same genus and abundant lichen and other species which are very minor quantities, and identify the compounds and isolate the compounds from the abundant species, and all these compounds were taken as a standards to map these compounds and their presence in the other species. So these are the compounds isolated from the Permotrema tinctorum, which is an abundant lichen, and based on this we have developed a total ion chromatogram based on the LCM, starting from sample preparation, optimization, and extraction optimization, and the conditions of chromatographic conditions, mass spectrometric conditions optimized, and then finally we have developed the total ion chromatogram of 5 species. Permotrema belongs to Permotrema genus, and identified, and based on their mass fragmentation pathways, identify the several molecules of different class of compounds, dipsidones, dipsides, paraconic acids, monophenolic, all these compounds, and based on the mass fragmentation of pathway, we have assigned the different class of compounds and also proposed the mass fragmentation. And these are the other compounds, and these are the dipsidones compounds, and the fragmentation pathway of other compounds, monophenolic compounds and paraconic acids. And finally, we also developed a Venn diagram, concluded based on the presence of the specific species or availability in all the species. Some of the compounds are available or present, identified their presence in all the species. Some of them compounds are specific to their only specific species. So similarly, these are other type of work also same. We have identified the different limonoids from different species, Malaceae species, and Malaceae plants by using the LCMS pathway, and starting from ethyl extract and SP cartridge mass spectrometer, developed the total ion chromatogram, and based on the mass fragmentation pathway, identified different molecules. Here, recent work, we also identified, isolated different compounds in the plant called Coccolocystis, very good antiviral medicinal plants, and the large high yielding compounds chinococcolin, magniflorin, and these 4 compounds we have quantified by using the MRM assay after developing the different and after optimizing the different conditions starting from extraction and identification. We have quantified further also, we have proposed the fragmentation pathway and identify some other molecules in the extracts. So I just to go to some of the aspects of quality control of herbal drugs, where the recent scenario, it is most important because based on the popularity of the herbal drugs, majority of the vendors are doing adulteration, which causes the genotoxicity or toxicity, which finally leads to the death. This is one of the recent clipping where in the August 1st, 23, where herbal supplement toxicity lands patient in the hospital. Because there are different problems in the Indian herbal industry, because presence of different regulatory authorities, regulatory requirement, and the limited market foreign countries are major hindrance for exploring, because we have different regulating authorities, and because we will almost we go the authority where the approval process is very simple, or we always follow the loopholes to get the compound or product approved without any standardization. A standardization and quality control of raw material and herbal formulation emerged as a major challenge in the herbal drug manufacturing firm, as the availability of the marker compound is becoming difficult. And in such cases, non-availability of regulatory guidelines to control herbal drugs, in case available guidelines are not implemented properly, good manufacturing practice, good agriculture and collection practices, the poor implementation of cosmetics in 1940s considered as a major hindrance in the herbal drug industry. Nowadays, if the major compound or responsible compound is the plant is not available or scarcely available, no herbal industries are using the synonymous plant where the major compound or active compound or therapeutic marker are low in yield. In that case, the efficacy may be doubtful, and sometimes even there is a chance of getting toxicity also. So because, and also the plants which are used in our system of medicine, traditional medicine, 75 to 80% are traditional medicines are the wild source, where sometimes sustainable supply of these materials also difficult, and all these medicinal plants are cross-pollinated species, and these are also affected with some genetic factors, season, and also the height and temperature, and its the soil also responsible for the production of the secondary metabolites. And now we also have lack of consistency in the quality batch to batch in production of products based on the marker compound, therapeutic marker, and lack of well-defined and well-orchestrated SOP. So these are the different parameters: quantity, dosage, quality, adulteration, efficacy. And here these are all parameters should be well checked for the end product, and physical, chemical, biological, and botanical. In the botanical, microscopic and macroscopic examination should be thoroughly done to identify whether it is a proper plant or it's a synonymous plant of synonymous plant. And similarly, moisture content, extractive value, and it should be pesticidal residue, free of pesticidal residue, heavy metal residue, and microbial contamination should not be presented, and pharmacological evaluation should be done. So these are the different analytical techniques which are approved by the US FDA and USDA, HPLC, HPDLC, UPLC, LCMS, and GCMS. All this technique has their own significance in the characterization and standardization of the herbal formulations. So here I just one example. We are also working in this area. This plant is Sasure Lappa, which is very well known in the northern part of India, and part of the many herbal formulations. And this what we have done, we have procured this material and quantified these 2 marker, dihydrocosis lactone and costonolite. So these 2 are the marker compounds, and we have quantified these 2 markers by developing the HPLC method and HPDLC method. And further, we purchased some of the products available in the market where this plant is one of the ingredient in those medicines, and whether we have checked whether these markers are in the detection limit and non-detection limit. Fortunately, majority of the drugs containing the marker compounds in the detection limit. Further, we also studied their stability, because the active or therapeutic marker is not stable in our intestine, where our intestine contains the high acidic environment. So if it is converting to something else, that new compound should not be toxic. Then only we will survive. So further, in that case, we also studied the degradation behavior, and in the stressful condition, fortunately these two compounds were stable enough. And this is another study, similar kind of study. We have done the analysis of the Boswellia serrata, Shallaki, and these 6 markers are well identified, well defined in different pharmacopoeia, and we also quantified the 6 compound in the raw material, and also developed a quantum total ion chromatogram, and also analyzed almost 20 to 30 marketed samples for the presence of the 6 markers, and almost very few industries or few samples are containing the 6 markers. So I conclude here. Once again, I would like to thank my director for his constant encouragement, and my supervisor, Dr. J.M. Rao, for training, giving training in this unique field of natural products, and of course, not the least, my students for their hard work, and also funding agencies, DST, DBT, ICAR, and then GSR for financial support. So here, these are the Indian pioneered scientists who contributed their entire life in the cherishment of natural products, starting from J.L. Simonson, Ramnath Chopra, Shalamuddin Siddiqui, T.R. Sheshadri, Venkatraman, T.R. Govindacharya, Asima Chatterjee, Susanka Chandra Bhattacharya, and Sukhdev. Sukhdev is called as a living legend of natural products, because this year he is celebrating his 100 years birthday. He is the father of glucosterones. Similarly, Asima Chatterjee is the first woman scientist who received the Bhatnagar Prize in India, and she worked on the alkaloids, majority of the work. And T.R. Govindacharya is a well-known person. He almost his entire research based on the neem plant. Similarly, other scientists also contributed a lot in the cherishment of the natural product research. And I'm happy to announce that, happy to share that 2 of my patents were commercialized to Sami Labs, and now that has been turned into a product, and now it is they have released this product in the international market based on our the patents. And these are some of the molecules present, and we are also working on some nutraceuticals where making the process by using the traditional medicinal food plant. So as part, we have developed the 2 process technologies, one for anemia and for combating the management of the diabetes, and we have commercialized to Amaya Life Sciences, and they made some work, and then they made a product out of it in the name of Superfoods to beat sugars, and this was released. The product was released in the March 2023 by the Honorable Science and Technology Minister from IICT. So now these products are available in Amazon and Flipkart in the name of Superfoods for Diabetes. In this, thank you once again for giving the opportunity to share some of our recent work and to share some of our recent views and views of the natural product drug discovery. And thank you once again for your patience.
Thank you, sir. Thank you, sir, Dr. K. Suresh Babu, sir.
Yeah.
Your presentation has given a lot of information who are going to work in the area of natural products and information regarding the various approaches using for the identification or isolation of the bioactive leaves from natural source and their derivatization. Thank you so much, sir. Now it is,
no, ma'am. I gave yesterday. I gave, so I don't know.
now this time is
They have kept inside. She was telling. She was cleaning, so she kept inside. I'll ask. Auntie, sign ಆಯ್ತಾ ಡೈರಿ
Suresh, sir.
Yeah.
ಆಗಿದೆಯಲ್ಲ ಆದ್ರೆ
some of the participants posting their questions.
Yeah, please.
Sir, how do we get marine products from research?
Oh, sorry,
How do we get marine products for research?
marine, there are, how do we get marine products for research? Because nowadays there is a lot of demand for products from seaweeds, marine algae. And of course, marine sponges and marine flora is also available, since our India has almost 9,000 kilometers of coastal region. And when coming to Andhra Pradesh, we have almost 900 kilometers of coastal region, which has been not explored the marine sources. And now government is focusing the fishermen to take a cultivation of marine seaweeds, because seaweeds are very much useful and which will give the income to the fishermen. Because in the Tamil Nadu, you can see a lot of industries based on the seaweed. Agar agar is the one of the best product derived from the marine seaweed. Similarly, there are some green seaweeds, green algae, which is a very good source of protein, where it is consumed as a food product in the different countries like Japan as a salad. And also, agar agar is one of the best product, and biofertilizer is also, there are some industries in Tamil Nadu, they are making biofertilizers from the different seaweeds. And cultivation practice, government is giving good training, free training for the fishermen who will come forward for their cultivation without any investment. That is also one of the biggest advantage of seaweeds or marine source.
Thank you, sir. So from my side, one question is there, sir. So what are the common problems encountering during the derivatization of the naturally aspirated compounds?
Yeah, because derivatization is a good aspect for improving the activity as well as the reducing the toxicity, as majority of the times pure natural products may not having the activity up to the mark. So that's what the majority of the natural product derivatives are became a drugs in the, if you see in the market. So when you are working on the derivatization, we should always go with the compound natural products. It's not like a synthetic compound, but we should identify some of the pharmacophores from the literature to attach that active pharmacophore to our natural products. So then only we will get the activity.
And by isolation, we are getting a very less amount of the product, right?
Yeah.
But there is any strategy to select the compounds for the derivatization?
Yeah, some plants will give two compounds in the high yield, some and other compounds are in the low yield. If you take the more amount of the raw material, then you may get you may isolate the compound in good quantities.
And our participant Rajendra asked, do we have any techniques to find before extraction of phytoconstituents?
How do you identify before extraction? It is not possible. It's not you go for X-ray or identify the compound. It's not possible. After extraction only, secondary metabolites will come into the solvent. Then you can identify the constituents and how many number of compounds by using thin layer chromatography or HPLC. Without doing extraction, how can you do? Only thing is you need to carry X-ray machine to the plant, and then you can see if the technique is not available so far.
Can you give me the strategy to select the solvents for isolation of selective phytochemicals? Like
Yes. You see, selection of solvent is also kind of a separation process, because if you start the extraction with non-polar, all the non-polar compounds and oils and fatty acids will come into the extract, and then go for the medium polar compounds will come in the extract, and finally go for polar methanolic extract and 10% water methanol and all glycosides, all polar compounds into the extract. So this is a kind of separation. So if you do directly methanol, you may miss some of the compounds which are soluble in hexane and medium polar.
Sir, for establishing the structural activity relationship, we are going for the derivatization. And
Yes.
whenever we are putting reaction, we are going for the reaction. the final compound completely depend upon the parent compound.
Yes.
Yeah, and maximum isolated compounds are cyclic in nature, majority of compounds.
Yeah, actually, structure-activity relationship, you can identify and isolate a many number of compounds, and after the getting the biological activity, you just analyze the compound. What is the major substrate is contributing the activity? Based on that, in the second generation, you can keep intact of that major functionality and then go for another derivatives. So in that case, you will identify the active pharmacophore, which is majorly responsible for the therapeutic. So that's what, if you see azole derivatives, antifungal drugs, starting from fluconazole to pasoconazole, rauconazole, majority of the drugs has been discovered based on the SCR relationship.
Sir, Mukta, participant asking one question: is there any analytical techniques for the characterization or isolation of fatty acids?
Yeah, fatty acids are characterized by using the GCMS, where those are called volatile, if they are a volatile compounds, and also you can also characterize it by even column chromatography also, isolated by column chromatography.
Thank you, sir. Thank you for your neat elaboration for the participant questions.
Yeah.
Thank you so much, sir. Though you are busy, you given valuable time.
Yeah, sorry, actually, this today 11 o'clock we have a divisional meeting. That's what I requested you to postpone to 2 o'clock. Thank you for that.
Thank you, Suresh sir. You are
Thank you, sir.
really enlightening the present students.
Thank
And
you.
really, we came to know that the depth of the discovery in the natural products, because
Yes,
we
sir.
well understood the objectives and scope of the natural products.
Yes,
Because
sir.
we are all working, but what is the depth and scope? Because once you're getting patent, that patent is getting marketized. Okay? These things we came to know that based on it, we well understood that how potential research will help to reach the market. That's what my takeaway points for this session. Thank you, sir. Dr. Suresh, sir.
Thank you, sir. Thank you.
I'm going to end this session. Thank you so much, sir, Suresh, sir.
Yeah, thank you, sir. So can I leave?
Sir,
It's my immense pleasure to vote of thanks for the session, sir, because you are really enlightening our students, and you are giving continuous support for us, so Vishnu College of Pharmacy. We are happy for that, and we will always be associated with you, sir, for coming research aspects as far as concerned. Thank you, sir. Thank you.
Thank you. Thank you, sir.